Greetings to all. I am Dr. Manonmani, Assistant Professor of Botany, Holy Cross College, Trichy. So now I am going to discuss you about the topic protein synthesis in plants, that is translation. So these are the learning objectives coming under this topic. So normally in plants, the protein synthesis is under the direct control of a DNA. So from the DNA, RNA and the ribosome of the cell are involved in the synthesis of proteins. So according to central dogma of molecular biology, the basic mechanism of protein synthesis is that the DNA nucleates regulates the DNA and also it forms the assembly of amino acids on the ribosome to produce the polypeptide chain. Since DNA cannot move out of the nucleus, it sends the messenger RNA and produce the polypeptide chain like this. So in this, the mRNA forms a template on the ribosome. Another type of RNA, that is tRNA, also involved in the synthesis of protein, so that it assembles the amino acids on the ribosome to produce the polypeptide chain. So this mechanism, this may be briefly explained as replication, transcription, translation and a protein. So we all know about what is replication. So in this the DNA polymerase is involved. So next coming to transcription. So normally in transcription is the copying of complementary mRNA from the DNA strand. In translation, the message present in the mRNA may be transferred into a protein. So that is a protein synthesis. So normally in the transcriptions, a portion of DNA which is to be transcribed first should unwind. So that the two enzymes, that is helicase and topoisomerase, these two are involved in the separation of two strands. So after the separation, one of the strand is involved in the synthesis of a complementary mRNA strand, so that it produces what? Adenine, guanine and cytosine, thymine, so likewise. Then so like this, so this is the DNA strand. So this may produce the complementary strand of a mRNA. So in this process, that means the process of a synthesis of mRNA, otherwise the RNA is called the transcription. So in this process, the enzyme involved is a RNA polymerase. Then in the genetic code, so normally the message present in the mRNA is form of a genetic sequence. So normally the nucleotide sequence, the three nucleotide sequence together called codons. Each codon specify one amino acid. So for in a genetic code, 64 codons are available. Out of 64, only 61 codons specify amino acids. The remaining 3 do not specify any amino acid, so that it called a nonsense codon or terminator codon or stop codons. So in the nucleotide sequence of mRNA and also the nucleotide sequence of tRNA, both are complementary with each other. So the triplet codes, that means the anticodon is also present in the tRNA. So the pairing takes place between the codon and the anticodon. So here codon is a initiation codon of a mRNA and anticodon is the tRNA. So the pairing takes place like this, A, U and GC like this. So the initiation codon may, if it may be a UU means so that may be paired with the anticodon AAA. So like this the pairing takes place. So the codons which do not produce amino acids means that is known as a nonsense codons. So this we all know about. Then the that is the codon. All the codons specify the amino acid means that may be called a codon dictionary. So after the synthesis of mRNA, what happened means that may be released from the nucleus into the cytoplasm, where it gets attaches to the smaller subunit of a ribosome. So after that it combines with the larger subunit also, so that it forms the polyribosomes during the initiation complex. So coming to the process, so it has 5 steps, that is activation of amino acids, then transfer of activated amino acids to tRNA, then initiation, elongation and termination. So first we'll see about activation of amino acid. So before the amino acid get themselves attached to tRNA, they have to be activated, otherwise energized. So that activation may be carried out with the help of an enzyme, amino acid synthetase. So it also needs magnesium divalent cations and also ATP. So these two are needed. Then after that what happened means this may produce what? Enzyme amino acid AMP complex. So during this process the pyrophosphates are released. So after the activation, the transfer of activated amino acids to tRNA. So the enzyme amino acyl complex reacts with tRNA so that the amino acids binds with the tRNA. So during this process what happens means the AMP and also the enzymes, so both are liberated. So this enzyme, that is amino acyl synthetase, has the double specificity so that it binds with a particular amino acid and then to a particular tRNA. So thus specificity of a particular tRNA picks up a particular amino acid. So this may takes place through this enzyme only. So for 20 amino acids, at least 20 enzymes are available. So similarly tRNA also has various anticodons to pick up these amino acids. So after that initiation, so it has two steps. So that is preparation of site and another one is initiation. So normally site means ribosome in cytoplasm. So these are the actual site of a protein synthesis. So normally the ribosome in cytoplasm are separated into their subunits. So that may be 50S and 30S subunits. So when it is not participating in a protein synthesis, it is remain as a subunit only. So during that initiation, what happen means the mRNA attaches with the 30S, that is smaller subunit of a ribosome. So through the AUG, that means initiation codon. So during this process, what happened? The initiation factors are involved because this only initiate the process. So the starting amino acid present in the polypeptide chain of eukaryote is a methionine. So in case of a prokaryote is a N-formyl methionine. So next the tRNA attaches with the amino acid. What happens? So that also combines with the 30S mRNA complex. So this forms the 30S initiation complex first. So after that 30S again combines with the 50S subunits, then finally it forms the 70S subunit of a ribosome. So this is called now initiation complex. So next is the elongation process. So after the initiation, elongation takes place. So in this, the 70S mRNA, then N-formyl methionine tRNA complex is involved in the elongation process. So like initiation factors are involved. In elongation also, elongation factor are involved, that is elongation factor T and elongation factor G. Then T is required for the binding of amino acyl tRNA to the ribosome. Then G is involved in the translocation process. So first is the binding of amino acyl tRNA. So this, as I told, so this is used to binding the amino acyl tRNA to the A site of a ribosome. Then coming to the peptide bond. So in between translocation and the first step, there will be peptide bond takes place. So that peptide bond is formed between the carboxyl group of the first formed amino acid and also the amino group of a second formed amino acid. So during this reaction, the peptidyl transferase are catalyzed. So like this, the elongation process takes place. So the next is translocation. So translocation is nothing but the movement of ribosome relative to mRNA. So during this process, what happened? The ribosome move along the mRNA a codon at a time from 5' to 3' direction. So and also from A site to P site. So this needs, as I told, the elongation factor G and also GTP. So like this, the elongation takes place. Then coming to the final step, that is termination. So as I told, out of 64, the 3 codons are called nonsense codon. These are UAA, UAG and UGA. So these 3 are called nonsense codon. So whenever the ribosome receives these codons, it receives the signals to stop further elongation and also terminate the process of a synthesis of polypeptide chain, so that it needs release factors, that is release factor 1, 2 and 3. So in this, the release factor 1 and 2 is involved in the recognition of the termination codons, and 3 alone is involved in the release of polypeptide chain, that means the completed polypeptide chain from its attachment. So that's all about the process, that means termination process. So this is the newly synthesized proteins. So come to conclusion. So RNA normally instruct the DNA to produce the protein. So these we all the DNA synthesis takes place inside the nucleus, then it sends the messenger RNA from the nucleus towards the cytoplasm. So in the cytoplasm, translation process takes place. So that's all about the translation. So these are the frequently asked questions, then references. Hope you all understood very well. Thank you.